The American cranberry is a native crop which has been cultivated in the U.S. for a little over 200 years. In 2023 alone, upwards of 8 million barrels of cranberries were produced in the United States, with a production value of nearly $35 million. However, with the increased restrictions on pesticide usage since the early 2000s due to environmental safety reasons, cranberry bogs are now seeing the resurgence of one of their most dangerous pests, the blunt-nosed leafhopper.
My name is Peter Outemans. I am a plant pathologist here at the Blueberry Cranberry Research Center in Chatsworth, New Jersey. Specifically with false blossom, it's an old disease that disappeared and is re-emerging. It's a phytoplasma, it's vectored by a leafhopper, and it has its own set of challenges to control and to manage. And so right now, we're just in the sort of figuring out stage to understand how to manage this disease. Historically, plant breeding was the way to go. But that's a long-term solution. And we have a lot of plants in the ground right now that are susceptible, and we need to figure out short-term goals to control that disease now.
Okay, my name is Cesar Rodriguez Saona. I'm an extension specialist in entomology at Rutgers University. I work on cranberries and blueberries, looking at insect pest management. So in my lab, we work on insect communication. So we study how insect interacts with other organisms through chemicals. So plants produce a lot of chemicals like volatiles that attract or repel insects. So we study how those chemicals can be used to control insect pests. We also look at how these chemicals are involved in trophic interactions, so not just the effects on the herbivores, but also on the natural enemies of the herbivores.
So we're studying here the interactions among the cranberry plants, the phytoplasma that causes the cranberry false blossom, and the herbivores that feed on the plants. We're looking both at the vector of the disease, the blind-nosed leafhoppers, but also we're looking at the effects on non-vectors. And we're also studying above ground and below ground herbivores.
So one of the things that we want to understand are the mechanisms associated with how the phytoplasma manipulates the host for its own benefit, but also for the benefit of the herbivores that feed on the plants. So by doing that, we could manipulate these responses to reduce the level of infection, or also reduce the traction, or increase the resistance to the herbivores that feed on the plants.
I have currently a student that is also looking at how nutrient availability in the form of fertilizer affects these interactions.
In the future, we want to also understand where the blind-nosed leafhoppers are coming from. One of the ideas behind it is that they are using alternative hosts in the understory of the forest, and that could be a source of infestation. So we want to see how the landscape is influencing the abundance of the blind-nosed leafhoppers in cranberry beds throughout the state.
Okay, so these are the latest generation of our crosses in the breeding program. We select for lots of different traits, including fruit yield and quality and potential disease resistance to many of the diseases that impact cranberry. Many of the crosses we make are using wild material or unimproved germplasm, and so they're less elite than some of the populations that are developed by our cultivar development program, but we're interested in using this unadapted or unimproved germplasm because it may contain unique traits that are absent in elite germplasm and that we want to move into elite germplasm. So we'll use these populations to screen for useful genetic variation and then from here we'll make selections that hopefully will advance and release to the cultivar development breeders so that they can use those as parents in their crosses and integrate the traits that we've discovered into their own breeding material. We're interested in screening diverse populations of cranberry for resistance to false blossom, either through resistance to the insect vector, blunt-nosed leafhopper, or direct resistance itself to the phytoplasma that causes false blossom.
I'm James Polishok. I'm a research plant pathologist with the USDA ARS, headquartered out of Beltsville, Maryland, and based in Chatsworth, New Jersey. My lab focuses on plant pathogen interactions, understanding plant diseases, and how we can better control them. So in cranberry false blossom, we're trying to understand how the disease is vectored and what insects can vector the disease. We're looking at alternate hosts of the disease and trying to understand where the disease is coming from and how it enters cranberry beds. We're also trying to understand how or why certain varieties seem to be more resistant to the disease and the genetic basis for that variation. Short term, we're looking at whether or not there's strain variation in the pathogen and trying to understand if strain variation influences symptom development and also what varieties of cranberry can be infected. You know, different strains can have different host range within cranberry varieties. So this is one of the methods we use to detect the phytoplasma. This machine is called a real-time PCR machine. It has the ability to read the fluorescence in reactions. The type of reaction we do is called LAMP, which stands for loop-mediated isothermal amplification. When we get a positive reaction, the fluorescent signal is read on this screen. These are our negative samples down here, and these are our positive samples. So we can use this assay to detect the phytoplasma in plants as well as insects. And we can also, by the amount of fluorescence, get a rough idea of the volume or the quantity of the relative quantity of the phytoplasma in the plant or the insect. So we can tell if there are differences in particular varieties of plants or even the types of insects that are potentially vectoring the disease.